Your brain may be lying to you, and science just proved it. In this video, we break down 10 mind-blowing takeaways from a shocking StarTalk episode with a Stanford neuroscientist who's literally building brain circuits in a dish. One of these discoveries could change how we treat pain, emotion, and even mental illness forever. Don't blink. Takeaway 1. Brain cells organize themselves like they know what to do. One of the most surprising discoveries in neuroscience is that human brain cells, when grown in the lab, do not need external guidance to know what to do. They self-organize into structures that resemble real brain circuits, much like a group of strangers finding their places in a large, unfamiliar building without a map. Sergiu Pașca explains that once the right types of brain cells are made in the lab, they automatically begin connecting to one another and forming structured layers, much like how they behave in the womb during natural development. Scientists initially believed they needed to provide detailed instructions to make circuits, but they found out the cells already carry that blueprint. He said once you make a specific cell, it actually comes with the instructions. The cells even trigger new instructions in neighboring cells when they interact, creating a ripple of organization. This means the lab-grown systems are not random at all. They are biologically programmed to build brain circuits just like in nature. For example, researchers put together cells from different brain regions, and inhibitory neurons move toward the correct destination, just like they do inside a developing human fetus, jumping in coordinated directions and timing without being told what to do. Now let's move to the next takeaway. Takeaway 2, stem cells can be made from skin. Stem cells are not just harvested from embryos anymore. Nearly 2 decades ago, scientists discovered how to take adult cells, like skin cells, and reprogram them back into stem cells. This process is like turning back the biological clock, making a mature cell forget its identity and return to a flexible state. These lab-made stem cells are called induced pluripotent stem cells. Pluripotent means they can turn into any other cell type, such as a brain, heart, or liver cell. This breakthrough eliminated many of the ethical problems surrounding embryo use and gave scientists access to stem cells from anyone. The process involves inserting a specific combination of genes into a skin cell, which confuses the cell into thinking it is young and undifferentiated again. For example, one researcher inserted just 4 specific genes into adult skin cells and successfully reprogrammed them into cells that behave like embryonic stem cells. These reprogrammed cells can then be grown into neurons in a dish, allowing researchers to study neurological diseases in a way that was never possible before. Now let's move to the next takeaway. Takeaway 3, real brain diseases can be modeled in dishes. The lab-grown neurons are not just for decoration. Scientists are now using them to study serious brain disorders like autism, schizophrenia, and epilepsy. These diseases are often too complex to be studied in animals like mice, which do not show the same behaviors as humans. Human brain tissue is also impossible to access for obvious reasons. So these models, called assembloids, give researchers a unique window into what is going wrong in the brain. Pașca started this work with patients who had severe autism-related conditions like Timothy syndrome, which includes epilepsy and heart problems. By turning their skin cells into brain cells, he could watch how those diseased neurons behaved in the lab. For example, his team found that a single-letter mutation in a gene caused a calcium channel to stay open too long, which disrupted the cell's communication with other neurons. That insight took over a decade to achieve, but would have been impossible without using the patient's own cells in this way. Now let's move to the next takeaway. Takeaway 4, drug testing on avatar brains is already here. One of the most promising uses of assembloids is drug testing. Instead of testing new drugs on animals or going directly into expensive and risky clinical trials, scientists can now test them on personalized brain avatars made from a patient's own cells. These assembloids mimic parts of the human brain and allow researchers to observe how drugs affect human neurons in a realistic setting. Pașca's team took brain cells with Timothy syndrome mutations, identified a potential treatment using molecular analysis, and then tested that treatment directly on the diseased assembloids. The treatment corrected nearly every defect in the neurons. To go a step further, they transplanted these human brain assembloids into rats. These rats then developed partial human brain tissue, which allowed them to test how the treatment would work in a living system. For example, They injected the same drug into the rat's bloodstream, and it helped the human brain tissue inside without causing damage. This is the future of precision medicine. Now let's move to the next takeaway. Takeaway 5. Some cells are programmed to feel pain. Scientists have now built a lab-grown sensory circuit that mimics the way we feel physical sensations, including pain. This involves combining 4 different types of organoids. One for sensory neurons that detect stimuli, another for spinal cord processing, a third for the thalamus in the center of the brain, and the last one for the cortex, where we become aware of the sensation. When combined, these cells began to synchronize and send signals down a unidirectional path as if they were practicing the feeling of touch or pain before any real stimulus was introduced. This model helps researchers understand why some people are hypersensitive to pain and others feel nothing at all. For example, when the scientists used CRISPR gene editing to introduce real human pain mutations into these cells, they observed fast, intense firing in the hypersensitive ones and total disorganization in the ones that could not feel pain, even though the circuits still looked normal. We're halfway through the video. Thanks for sticking with us. If you're enjoying it, hit the thumbs up and share it in your WhatsApp groups. If you'd like to support us, please tap the thanks button below. It helps us keep making great content. Drop a comment and don't forget to hit subscribe for more. Now let's continue with the video. Takeaway 6. These circuits track time without a clock. A surprising feature of brain cells in a dish is that they can keep track of time even without a body. These cells progress through development just as they would inside a fetus, hitting milestones after certain numbers of days. Researchers found that even without exposure to the outside world, the cells began to resemble a postnatal brain after about 9 months in a dish. This means the cells have an internal timer or clock that tells them when to change, despite not seeing light, hearing sound, or being part of a living person. For example, when scientists cultured brain organoids made from chimpanzee and mouse cells, each developed according to their species' normal gestation period. The chimp organoids developed in about 8 months, while mouse ones took just a few weeks, showing that the clock is species-specific and coded into the cells themselves. Now let's move to the next takeaway. Takeaway 7. These models challenge our idea of intelligence. As the lab-grown brain systems get more complex, scientists are starting to ask difficult questions about ethics and consciousness. The assembloids do not currently think, feel, or learn, but they do coordinate and respond to stimuli in ways that hint at the beginning of more advanced behaviors. Pașca emphasizes that the goal is not to create intelligent brains, but to make useful models. Still, the line gets blurry. At what point does a system become advanced enough that it deserves protection or rights? This is not just science fiction. For example, these models already allow researchers to observe the beginnings of coordination and rhythmic firing, which is how real brains prepare for future sensory experiences. This could open the door to uncomfortable questions, such as whether it would be ethical to destroy or experiment on such a system once it gets complex enough to learn or remember. Now let's move to the next takeaway. Takeaway 8, ethics can't be ignored anymore. The more human-like these brain models become, the more ethical issues arise. First, there is the question of consent. Many of these cells come from patients or donors, and researchers must be clear about what will be done with them, including whether they will be transplanted into animals. There is also the issue of ownership. Who owns the cells once they are grown into organoids? There is also concern about animals used in transplantation experiments. Are they suffering if they have human brain cells inside them? Finally, there is the deeper question of whether we should be doing this at all. Pașca says that psychiatric diseases affect 1 in 4 people and cause enormous suffering, so ignoring tools that could help seems irresponsible. Still, he and others in the field are now bringing in philosophers and religious leaders to help guide decisions. For example, a historic meeting was held in California, the same place where gene cloning ethics were debated in the 1970s, to begin shaping regulations around this new frontier. Now let's move to the next takeaway. Takeaway 9, neurodiversity versus treating profound conditions. A growing movement promotes neurodiversity, which sees conditions like autism not as diseases, but as natural variations of the human mind. However, Pașca is quick to point out that the patients he is working with suffer from profound autism, which is not the same as simply being quirky or introverted. These patients cannot speak, make eye contact, or function independently. Some experience dozens of seizures a day. While society debates where the line is between normal and abnormal, these families live with the harsh reality of extreme disability. Pașca says he is not trying to erase uniqueness, but to help those with severe suffering. For example, he mentions parents whose biggest fear is dying because their child will never be able to live alone or care for themselves. The focus is on giving these children a chance at a normal life, not making everyone conform to one definition of a healthy brain. Now let's move to the last takeaway. Takeaway 10, we may never fully replicate the brain. Despite all the progress, there are serious limits to what these brain models can do. They are not vascularized, which means they have no blood supply. They also do not receive complex sensory input like a real fetus would. Sensory input like vision, sound, and touch shapes the brain in ways that cannot be replicated in a dish. Even small changes early in life, like covering a kitten's eye for a week, can cause permanent blindness. The assembloids are useful, but they will never be the real thing. As Pașca puts it, all models are wrong, but some are useful. The key is not to make a perfect brain in a dish, but to make good enough models of specific circuits that can lead to new treatments. For example, while we may never grow a fully functional brain in the lab, we might be able to use a simplified model of the spinal cord to study poliovirus and find a cure for paralysis. Thank you for watching. If you'd like to support this channel, hit the thanks button below. It really helps us keep going. If you enjoyed this summary, please leave a like and share it in your WhatsApp groups. To join discussion about this video, drop a comment below. And for more videos like this, hit the subscribe button below.